Hi guys, and welcome to Chapter 6. This will be interaction between cells and the extracellular environment. So you should find this PowerPoint in your Moodle. You can print it off before you go through here so you're able to take notes as you walk with me through this PowerPoint here. So the introduction, the extracellular environment includes everything located outside the cell. And so when we talk about intracellular, it's going to be everything inside the cell. And this PowerPoint is going to talk about how they communicate and how they move substances such as water and molecules throughout the cell. So 67% of our water is going to be found within the cells of our intracellular environment or compartment. 33% is in the extracellular compartment. 20% is in blood plasma. Now these percents are going to be in your test, so make sure that you know these numbers. Let's see if I can get all this in here. And 80% makes up what is called the tissue fluid or interstitial fluid. And that's what will connect the intracellular compartment to your blood plasma. All right, and how do we do that? So we have blood vessels located underneath the skin, and interstitial fluid is going to be coming out of the blood vessel into the surrounding tissue, and we always want to keep a perfect homeostasis or osmotic pressure with our blood vessels, and this is how we keep our blood pressure at an optimal number. So what is the normal blood pressure? Probably somewhere between 140 over 80. They have changed the numbers just a little bit in the nursing realm, so those are just the numbers that are roundabout somewhere between normal blood pressure. Alright, looking at a cell on the extracellular matrix, what does it contain? So the stuff in red here will be on your first exam, so make sure you go over this and make note of this, that they contain protein fibers of collagen and elastin and a gel-like ground substance. So they're going to support the cell, give it that shape that it needs, and keep it strong. Also located in the cell membrane to help keep the shape and keep your cell membrane strong, and to help things pass through here are something called integrins. So what are the functions of integrins? This will be on one of your worksheets, so you will need to know these three. And the first one is impart a polarity to cells, and the second one is affect adhesion and motility, so what things can attach and what can't, and affect proliferation, number 3 there. And so you want to make sure you know those three for your exam. Alright, so let's talk about the plasma membrane, the cell membrane, the cell wall. Remember, it has a lot of different names. So the plasma membrane, we say in green here on your test, it'll be selectively permeable. So that means it allows some molecules to cross, but not others. So it's generally not permeable to proteins, nucleic acids, or other large molecules. So if it's really, really large, it's going to be harder for substances to pass through. Generally permeable things is ions, nutrients, and waste, and so those things will easily pass through here. So now we'll go through the different types of membrane transport, and we do have simple diffusion, facilitated, primary active transport, and we'll talk about all these in great detail and what they mean. So simple diffusion, as you see on this PowerPoint slide here, the simple diffusion does not need energy and able to pass through the cell membrane quite easily. And remember, our cell membrane is made up of our phospholipid bilayer, or 2-part lipids, remember, with our heads and our 2 tails. And remember, the heads are hydrophilic and the tails were hydrophobic. So remember those things, as they will be on your exam as well. Alright, so categories of membrane transport. We have non-carrier versus carrier. So what does that mean? A carrier means, are we going to be able to use proteins to get things across, or can we just simply diffuse or go through the cell membrane without the help of a protein? And so these are what that means. And so for our non-carrier mediated, we have simple diffusion, and osmosis, and we'll go through these here in a minute, and our carrier mediated, which means we know we need a protein to help us out. We have facilitated diffusion, and we have active transport. And you might note that 1 through 3 up here, these non-carriers, these 3 items are passive transport items, and the ones below here are active. And active just means they need ATP, and the top 3 are passive and they don't. And so here they are one more time. What are our passive versus our active? So our passive has diffusion, which means no energy is required. We do have facilitated diffusion, which does not contain energy. It just has a protein to help it, but no energy. And our active transport needs, we need ATP, or we need energy. And so on your test, I may ask you, what's an example of an active transport? And you might put the sodium-potassium pump. So the sodium-potassium pump needs a great deal of energy to work. Also, endocytosis and exocytosis do as well, and we'll talk about each one of these in great detail. So, types of passive, nonpolar, channel proteins, carrier proteins, and if you think back from biology or the earlier chapters, even in this class, we talked about the 6 different types of proteins and their different functions. And so they're going to list some here that fall underneath the category of passive, once again meaning that they do not require ATP to work. And remember, what is a type of active transport? It's going to be sodium-potassium pump, as we're going to need a protein, which are these purple circles here. We need a protein in order to work, and we also need ATP in order to move sodium and potassium inside and outside the cell. And I have some videos in Moodle where you can watch each of these in detail if you wish. Alright, diffusion and osmosis. So first off, we have to start out about what is a solution. So when you'll be doing your experiments, you may throw these in your lab report as well. So a solution consists of a solvent and a solute. And molecules, remember, if you took chemistry class, are always in a constant state of motion. They're in green, they never stop. And they're always in a state of motion, which means they're always trying to reach what we call equilibrium. And this is going to obey the second law of thermodynamics. So diffusion of a solute. So we want to get our higher concentration and our lower concentration to equal out, or equilibrium as we call it. When you do your osmosis lab, you'll be dealing with a dialysis membrane and some iodine. And your dialysis tubing is going to act as your cell membrane in this case, and you'll be processing out simple diffusion, and you'll watch molecules move in and out of this dialysis membrane. And I can't tell you more about it, so you'll have to wait and do your lab and you'll see. Alright, diffusion through a plasma membrane. What are some things that pass easily? And for your test purposes, it'll be small, nonpolar, which means uncharged, lipids that are going to pass through there, such as molecules such as oxygen, carbon dioxide, and steroid hormones. So the things in blue on your PowerPoint here, if you can see, those are things that are going to pass easily through the cell membrane or your plasma membrane. And we do have gas exchange that happens at the cell. So we have CO2 and O2. And of course we want those to easily pass through the cell, so that they can always keep high oxygen in their tissue. And number 3, water can pass through easily, of course, using something called aquaporins. Now this may be a question on your homework, and your answer will be aquaporins, and these are just little tiny pores in the cell membrane that allow water to pass through there. Pretty easy. Alright, what are some things that are, don't pass through there very easily? We'd have to say larger polar molecules can not pass through the membrane by simple diffusion. So these things, if they're too big, as you see in this picture down here, this PowerPoint, if items are too big to pass through the cell membrane, they will in fact use a protein to help them through the lipid bilayer. Remember, what is osmosis? This is where water is moving, not molecules, but water, from an area of high concentration to an area of low concentration. What is tonicity? You'll have to know what this is for your upcoming exam. So tonicity takes into account the permeability of the membrane, and basically it's going to be how substances are moving in and out of this cell, and specifically they talk about red blood cells. So let's just take a look at these red blood cells here. So what is the fate of the red blood cell in an isotonic, hypotonic, and hypertonic solution? Well, isotonic, if you took MedTerm at all, or if you're going to, iso means the same. So this means we have equilibrium. Nothing is happening on the outside and inside of the cell. We are at equilibrium and there is no effect to the solution. In a hypotonic solution, we have water that is going to move inside the cell because there is a greater concentration of water on the outside. And where does it want to move? It wants to move inside. So remember, osmosis, an area of high concentration, wants to go to an area of low concentration. But what will that do? In turn, it will pump the cell up, and it could cause the cell to burst. Hypertonic is the opposite, and it means we have a lot of water inside the cell, not a lot of water outside. So where does it want to move? It wants to go from the inside of the cell to the outside, so it will cause it to shrink. And this other slide here depicts this pretty well. So make sure you know the difference between those three things. Alright, there is a transport across the cell membrane video. I encourage you to watch. It is on YouTube. Alright, what regulates our blood osmolality? So what regulates our blood pressure? And what it's going to be is our hypothalamus, which is located in our brain. And so I put it in here in green because it is going to be on your exam. So it's going to detect whether you're dehydrated or whether you're not dehydrated, and it will adjust your blood vessels to absorb or let go water as it needs in order to keep your osmolality perfect in homeostasis. Alright, carrier-mediated transport. So once again, if items are too large or they're polar and they can't go through the cell membrane, they need to have carrier proteins to help them to go through that, to help them do that. And also facilitated diffusion needs that as well. So no ATP is used, high to low. You probably went through some of these in high school here. This all talks about the sodium-potassium pump. I encourage you to watch this video on YouTube, and it goes very in depth about this and what it is, and it will make more sense to you hopefully. Bulk transport, when you talk about this, is